Okay, so starting out here, what we have is an immunoglobulin, same thing as an antibody. And immunoglobulins have a heavy chain expressed here with the dark blue, and they have light chain, or two heavy chains rather, with dark blue. These two heavy chains are bound together by a disulfide bond. Also, the light chain is bound to the heavy chain by a disulfide bond. On the heavy chain, there is one variable portion, and there are either 3 or 4 constant regions. So variable, constant, constant, constant. There's 3 if you're talking about the isotype IgG, IgA, or IgD, and the isotype of IgM and IgE has a 4th constant region shown here, C4. Down here on this whole portion here is called the FC portion, and the FC portion has it can bind to the membranes or be stuck in the membrane if it has a hydrophobic region in the middle here and a hydrophilic region here. It can stay membrane bound. These constant regions are true to their word. They're usually constant, somewhat constant, whereas these variable regions out here are much more variable. On the variable regions, expressed right here, you have 3 little loops on each one on the variable regions. The inner one here is CDR1, CDR2, CDR3. Same thing on the variable of the heavy chain, CDR1, CDR2, CDR3. And it's the CDR3 one that is the most variable, or it's also the one that's the most responsible for the binding. These light chains, depending on the person's genotype, can either be a kappa or a lambda. Typically, a person will have a certain amount of kappa and they'll have a certain amount of lambda in their body. Usually that ratio is about 60% kappa, about 40% lambda if in a normal person. But if someone develops a lymphoma where one of these becomes monoclonal, basically what will happen is one of these B cells will start kind of producing out of control, and it'll be either a kappa or it'll be a lambda, and so it'll produce tons and tons of kappa or tons and tons of lambda. And if you see that, if the ratio isn't 60-40, you see somebody has like a 90-10 or an 80-20 or something like that ratio, then you know that this person probably has lymphoma. I was talking about these different isotypes over here, but first I guess I'll discuss idiotypes. There are different idiotypes for these. Idiotypes are going to be what this portion binds to. Okay, so that's basically the variable region. If these things have a different variable region, then it's a different idiotype. You can have the same idiotype, but a different isotype. So you could have an IgG and an IgM that both have the same idiotype, but those are different isotypes. An isotype has to do with whether it's an IgG, an IgA, an IgM, or an IgE, or an IgD. Ig is immunoglobulin. And so those are usually found with, the differences are found in their basic structure. IgGs are monomers, IgMs are either monomers IgAs are either monomers or dimers, IgMs are pentamers, IgEs are monomers, and IgDs are monomers, but they're membrane bound and they're only found in naive B cells. Of the IgGs, there are 4 subtypes, 1 to 4 subtypes. Of the IgAs, there's 1 to 2 subtypes, and the other ones don't have any subtypes. You will find 13.5 milligrams per milliliter of IgG in the blood. You'll find 3.5 of
the same milligrams per milliliter, you'll find 1.5 in M, 3.5 is A, 1.5 is M, 0.05 is E, and D, you don't find any in the blood because they're always membrane bound to naive B cells. IgG is responsible for binding complement, and that is, it binds it with its Fc portion. It's also the one you'll find for neonatal immunity, and it also is involved in opsonization. IgA is involved in, it's in the mucosal tissue, so you find it in like breast milk and things like that. IgM is also responsible for complement activation and opsonization. IgE is involved with helminths and allergies, and IgD is what you find on a naive D cell. I'll explain that right now after I explain this real quickly. In the blood, typically, IgG will last 21 to 28 days. This is because of neonatal recycling. They call it FcRn recycling. Basically, it's where endothelial cell will take this guy in, and then he'll fix it up, and then he'll release it on the other side. And that's how it crosses through like the placenta and goes into the baby, goes into endothelial cells. IgA will last 3 days in the blood, or it has a half-life of 28 days. So this one has a half-life of 21 to 28 days, the G does. IgA has a half-life of 3 days. IgM has a half-life of 4 days, IgE 2, and IgD doesn't live in the blood. The IgA will last longer if it's in the milk or in the mucus or whatever. So if you have bottled milk, it'll last longer in the bottled milk. If we're talking now about this process of what these guys go through, typically they always start out the B cells start out as an IgD, and these ones are basically they're always going to be membrane bound, and once they become they go from being naive to being activated, they will have they'll start creating proteins that'll make IgM instead of IgD. And they'll do IgM for the first little while until they begin to age. And once they've aged a little bit, they'll start to produce A, G, or E. And then later they can differentiate into a plasma that pump out tons of antibodies, or into a memory that kind of stay constant in the body. If you wanted to create the like monoclonal antibodies, what you do is you would take a rat, inject it with something like measles, take the B cells that are producing antibodies that are going to help you out, take that B cell, combine it with a monoclonal immortal, basically cancerous cell from a human, and then combine the two, and it will make a B cell that is basically it'll make an immortal cell that's making antibodies. You can also do this by taking the DNA out of the B cell from the mouse and put it into a human cell, and that'll kind of do the same thing. It's more like, it's called cassette switching. Antigen is any molecule that can produce an immune response. Immugen is when it actually is producing an immune response. This is a humoral immunity response. It is producing a humoral immunity response. Hapten is where there's lots of small subunits. Like, I don't know if this example works, but let's just say you were to break up a bunch of lipopolysaccharide in your body, and here you have an antibody, and maybe one little piece of this lipopolysaccharide might not be enough to bind it, but if you get a bunch of these little haptens on there, it might be enough to actually activate an immune response. Then you have the epitope, and the epitope is the specific part of the antigen or immunogen or whatever that is the binding portion. Affinity versus avidity. Affinity is the strength of that single bond. So it'd be like how strongly this part fits onto your microbe or whatever. Avidity, going back to affinity, this is expressed in the dissociation constant, which is going to be the inverse of the strength. So you have a higher dissociation constant represents a smaller affinity, and vice versa. Then you have avidity, which is basically saying whether something is monovalent, bivalent, or multivalent. Remember I expressed earlier that there's IgG, it's a monomer, so it only has two little binding sites. A monovalent would be an example where only one of these guys is bound. Bivalent would be where you've got two bound. multivalent, where you have like somewhere between 3 and 10 bound. So that'd be like something like maybe an IgM. Then we have lastly the different types of ways these things combine. We have overlapping, where essentially one binding site overlaps with another binding site, or we can have non-overlapping, where one binding site doesn't overlap with another binding site. We can have linear, where maybe when the protein was in its fully like tertiary structure or conformation state, non-denatured, that couldn't bind, but once it's been denatured, it's a straight protein, then the antibody binds to it. Or you have the other one where it's conformational, which means that it only binds when it's in a non-denatured state, but when it's denatured, it doesn't bind because they're like in separate places, as I tried to show there. And that's most everything.